Welcome back, everyone, to the Deep Dive. Today, we're going to take a look at some of the most interesting stories making the rounds in health and tech. We've got a lot to cover today, from potential issues with inflammation drugs to some really amazing advancements in RNA sequencing. So let's dive in.
It's interesting, actually, how much of today's news really touches on the same theme. Like, we are getting so much better at understanding the tiny details of how life and disease work, at a molecular level. It's kind of blowing my mind.
Yeah, absolutely. I mean, look at this report from News Medical. Understanding how JK inhibitors may increase blood clot risk. This is a pretty big deal, because a lot of people rely on these inflammation drugs. So why are we seeing this potential clot risk?
Well, think about it this way. JK inhibitors work by targeting specific pathways that are involved in causing inflammation. But, and here's the thing, these pathways, they aren't only involved in inflammation, they're also doing other important things in the body, like helping with blood clotting. So by blocking them, you could be messing with this delicate balance, and in some people, that might lead to a higher chance of clots.
It's so complex how it's all connected, isn't it?
It really is.
Okay, so let's move on to something a bit more positive. RNA sequencing has been making huge strides lately. I was looking at the RNA-Seq blog, and they have a bunch of updates. One that really caught my eye was this CNET integrating protein interaction networks with RNA sequencing. It sounds pretty technical, but it seems like a game changer for understanding how genes and proteins actually work together.
Oh yeah, this SNET thing is really cool. You see, RNA sequencing can tell us which genes are active, like which ones are being expressed in a cell, and how much. It's like seeing which instruments are playing in an orchestra and how loud. But SNETnet takes it a step further by adding information about how the proteins made from those genes are interacting with each other. So now we have a map, not just of the instruments, but of the musicians and who they're playing with.
Wow, that's a great analogy. It really makes it clear how this adds a whole other layer of understanding.
Right. And this kind of detailed view can help us figure out how disruptions in these networks can cause disease. It's much more powerful than looking at just RNA or protein interactions alone.
Okay, so another fascinating one from the RNA-Seq blog. SL SmartC CLIP3 Express RNA sequencing reveals mechanisms of antigenic variation in trypanosomes. Now, these trypanosomes, they're nasty parasites that cause things like sleeping sickness. And this research used RNA sequencing to uncover how they actually manage to evade our immune systems. Talk about a sneaky enemy.
They really are masters of disguise, These trypanosomes, they constantly change the proteins on their surface, like they're wearing different masks, and this makes it super hard for our immune system to recognize and attack them.
Right.
So what this research did was use a really advanced RNA sequencing method to pinpoint exactly which genes are responsible for these changes. It's like figuring out the enemy's playbook.
No.
And once we know how they're switching things up, we can start developing ways to target them more effectively and hopefully come up with new treatments.
All right, so let's shift gears a bit and talk about something with pretty direct implications for genetic disorders. There's an article titled RNA Sequencing Reveals Novel Gene Regulatory Mechanisms in Dravet Syndrome. Now, Dravet syndrome is a really severe form of epilepsy, and this research has pinpointed these things called poison exons as a key factor. Could you explain what these are?
Sure. So, you know how genes are basically like recipes for making proteins, and exons are like the important ingredients in those recipes. Well, sometimes during this process of making a protein, there are bits of genetic material called introns that need to be removed, like taking out the seeds from a fruit before you eat it. But sometimes these poison exons, they sneak in and mess things up.
Okay, I see.
They're like rogue ingredients that shouldn't be there. They can make the protein not work properly or even become harmful. And in Dravet syndrome, this happens in a really critical gene called SCN1A. And RNA sequencing has been able to identify exactly where these mistakes are happening, which gives us hope for finding new ways to correct them and potentially treat the condition.
That's incredibly promising. So let's talk about the more technical aspects of RNA sequencing. The RNA-Seq blog highlighted this new innovative bead design that will enhance single-cell transcriptomics accuracy. It seems a bit specific, but getting these research tools to be more accurate is incredibly important, right?
Absolutely. Single-cell transcriptomics lets us look at the gene activity in individual cells, which is amazing for understanding complex systems. But the process involves a lot of very precise steps, and if there are errors early on, they can really mess up the results.
Right.
This new bead design focuses on minimizing errors during a crucial step where DNA is synthesized, basically copied. By making sure this step is more accurate, we can be much more confident in the data and in the conclusions we draw about how cells work and what goes wrong in disease. It's like making sure the foundation of a building is strong.
Makes sense. Okay, so the last RNA sequencing update we have is a really interesting one. Researchers use RNA sequencing to compare pig and human retinal organoids for vision restoration. This is really cutting-edge stuff.
It really is. Retinal organoids are these amazing mini retinas that scientists grow in the lab from stem cells. They're incredibly valuable for studying how the eye develops and what happens in diseases.
Wow.
And by using RNA sequencing to compare the gene activity in pig and human retinal organoids, researchers can see how similar they are and where they differ. This is important for knowing if pig organoids are a good model for human eye conditions, and also for figuring out how to use these organoids to develop cell-based therapies to restore vision in people who have lost it. It's a really exciting step towards potentially reversing blindness.
It really is incredible. And speaking of this technology moving into clinical use, we have news that Genodex, a major company in genetic testing, will be presenting their findings from their RNA sequencing program at an upcoming genetics meeting.
Yeah, this is another sign that RNA sequencing is becoming a standard tool in healthcare. Genodex focusing on this technology at a major genetics meeting shows just how valuable it is for diagnosing and understanding genetic disorders. It's not just for research anymore, it's moving into everyday clinical practice, which means we can get even more insights into how diseases work and potentially tailor treatments more effectively.
Absolutely. Okay, so now let's move on to a really fundamental challenge in genetics: understanding DNA methylation. The Omics blog called it Mission Impossible, Methylomics, and it highlights just how tricky it is to decipher these crucial epigenetic marks. For our listeners who might not be familiar, what exactly is DNA methylation and why is it so important?
Imagine our DNA as a giant instruction manual for building and running our bodies. DNA methylation is like adding little notes and highlights to that manual. It doesn't change the actual instructions, but it can affect how they're read and followed.
Okay, so it's like adding extra information on top of the DNA.
Precisely. It involves attaching a tiny chemical tag called a methyl group to certain parts of the DNA. These tags can turn genes on or off or fine-tune their activity, and this is incredibly important for all sorts of things, like how our cells develop, how they specialize into different types, and even for preventing diseases like cancer. Understanding these methylation patterns, especially in easy-to-access samples like blood, could be a game changer for detecting and monitoring diseases early on.
I see. So the blog pointed out that the best technologies for directly reading these methylation marks, without any extra steps, are from Oxford Nanopore and Pacific Biosciences. But even those have limitations, right?
Yes. While these technologies can directly see the methylation marks on individual DNA molecules, which is amazing, they need a lot of DNA to start with. And that's not always possible, especially when dealing with limited samples. Plus, using PCR to make more copies of the DNA, which is a common technique, actually erases these methylation marks. So it's a bit of a catch-22.
So it's a trade-off between being able to read long stretches of DNA and preserving these methylation marks.
Exactly.
So then there's the more common method using bisulfite, which the blog jokingly called a weapon of mass sequence destruction. Why is it called that, and what are the downsides?
Well, it's called that because bisulfite treatment is pretty harsh. It basically converts certain DNA bases into others, which lets us see where the methylation is. But the problem is it also scrambles some of the original DNA sequence information. It's like trying to read a book after someone's gone through and erased some of the letters. It makes it really hard to also look for genetic mutations in the same sample.
That makes sense. But the blog also mentioned some promising new approaches from companies like Watchmaker Genomics and Biomodal. What are they doing differently?
These companies are trying to be more precise and less destructive. Watchmaker has a method that only targets the methylated parts of the DNA, leaving the rest untouched. So you can still read the original sequence.
Oh, that's clever.
Right. And Biomodal uses a different approach. They make a copy of one strand of the DNA and then use special enzymes to distinguish between different types of modifications, including methylation. It's like having a backup copy of the book with notes on where the important highlights are.
So both of these approaches are trying to give us a more complete picture without messing up the original information.
Exactly. They're really important steps towards getting a clearer and more accurate view of the methylome.
Okay, now for a really intriguing development. Roche has this new sequencing technology called SBX. And the blog mentioned that it shouldn't be able to read methylation because it requires copying the DNA first. But apparently, Roche might have found a way around that.
Yeah, this was a bit of a surprise. SBX is a single molecule sequencing technology, but it copies the DNA before reading it, so everyone assumed methylation information would be lost in the process. But at a recent conference, a Roche scientist hinted at something called a fifth signal level in their data, which suggests they might have figured out how to detect methylation even after the copying.
Wow, that's fascinating. It sounds like they found a way to have their cake and eat it too.
Exactly. We don't know the details yet, but it's a sign that they're doing some really innovative work in this area.
So the blog talks about this impossible mission of making SBX directly read 5-methyl-C, which is the key mark of DNA methylation, possibly by using a third base pair. Is that even possible?
It's definitely a huge challenge. The idea is to expand the DNA code beyond the usual 4 letters A, C, G, and T and add a new artificial letter. Then they would need to find a way to convert 5-methyl-C into this new letter so that SBX can read it. It's like rewriting the DNA in a new language.
Okay, I think I'm starting to get it. And the blog mentioned 2 potential strategies,
Yeah.
using chemistry or protein engineering, but it also said that there are no known starting points for either. So it really is uncharted territory.
It is. No one has ever done this before, so it will require some truly groundbreaking research.
It really is mission impossible then.
It is, but the potential rewards are enormous. If we can crack this, it could revolutionize how we study and understand DNA methylation.
Absolutely.
Yeah.
So the blog also brought up the practical considerations, like how many steps are involved, what equipment you need, and the cost. These are all really important factors if we want this kind of technology to be widely used.
They are. A brilliant idea that's too complex or too expensive won't have much impact. For it to really change things, it needs to be practical and accessible to researchers and clinicians everywhere.
Okay, so let's move on to another exciting area. The integration of biological data with artificial intelligence, or AI. We have several articles from Nature that highlight how powerful this combination can be. It seems like AI is becoming essential in biological research.
It is. We are generating so much biological data these days, from DNA sequences to protein interactions to images of cells and tissues. It's simply too much for humans to analyze on their own. AI gives us the tools to sift through this data, find hidden patterns, and make new discoveries that we wouldn't be able to see otherwise.
It's like having a superpowered research assistant.
Exactly.
So several of these Nature articles talk about combining RNA sequencing data with protein-protein interaction networks. For example, there's a graph neural network that combines RNA-Seq and protein interaction data, and CNET, learning context-specific gene and cell embeddings by integrating single-cell gene expression data with protein interactions. What's the advantage of bringing these 2 types of data together?
It gives us a much more complete view of what's happening in a cell. Remember that analogy of the orchestra?
Yeah.
Well, this is like not only seeing which instruments are playing and how loud, but also how the musicians are interacting with each other, who's leading, who's following, and how the music is being created.
Right. It's like understanding the whole performance instead of just individual notes.
Exactly. And this can help us understand how networks of genes and proteins work together to control cellular processes and how those networks might be disrupted in disease.
Okay, so we're also seeing AI being used in a ton of different ways in biology and medicine. There are articles about analyzing images, making AI more transparent, and even using AI to triage skin lesions.
Yeah, AI is really versatile. It can be used to analyze all sorts of data, from microscopic images to patient records, and each of these applications has the potential to improve our understanding of biology and lead to better healthcare.
It's really exciting to see how AI is transforming the field. So there's one last example I wanted to highlight. It's an article called Neutrophil Elastase Binds at the Central Domain of Extracellular Toll-like Receptor 4, AI Prediction, Docking, and Validation in Disease Model. This is a case where AI is helping us understand how diseases work at a molecular level.
This is a really cool example of how AI can accelerate drug discovery. Toll-like receptor 4 is a really important protein in our immune system. It helps us detect and fight off infections, but when it's overactivated, it can cause inflammation and contribute to diseases.
Nice.
So understanding how other molecules interact with this receptor is crucial for developing new drugs. And in this study, researchers use AI to predict how a specific enzyme, neutrophil elastase, binds to this receptor, and then they confirm these predictions with experiments. This kind of information is incredibly valuable for designing drugs that can target this interaction and potentially treat inflammatory diseases.
Wow, that's amazing. Okay, so finally, let's talk about something that's really important for scientific progress. Data sharing. There's an article from BioIT World Flatiron Health enables patient-level data sharing across national borders. That highlights this.
This is a huge step forward, especially in the fight against cancer. Flatiron Health is making it possible for researchers in different countries to share anonymized data from electronic health records. And this kind of global collaboration is essential for making faster progress against complex diseases like cancer. By pooling data from diverse populations, we can get a much clearer picture of how cancer develops and spreads, and we can identify more effective treatments.
It makes so much sense to share data and collaborate on a global scale. It's the best way to accelerate progress and ultimately improve patient care.
I couldn't agree more.
So, that's a wrap for today's deep dive. We've covered a lot of ground, from the potential risks of inflammation drugs to the remarkable advancements in RNA sequencing and the transformative power of AI. It's an exciting time to be following these fields, and we can't wait to see what new discoveries emerge in the days and weeks to come. Thanks for joining us, and we'll be back tomorrow with more news from the cutting edge of health and technology research.